We showed that we can isolate and assemble probiotics that can both promote coral health and at the same time mitigate impacts from pollution, and that can contribute to a reef's persistence in a changing world.
The isolation and screening approach described can be used to select microorganisms presenting key metabolic activities, such as the degradation of pollutants or other beneficial characteristics to marine organisms such as corals.
That this method can be applied to isolate microorganisms able to degrade any contaminants, and additionally other sources of material can be used, any other environmental samples such as plants, soil, water, anything.
Study the literature and try to focus on strains that can be good candidates for your purpose. If possible, direct the isolation methods for those strains and avoid known pathogens. And if you are aiming to use a consortium of bacteria, it is also very important to first investigate if the members of the consortium have antagonistic activity against each other.
Because corals are under increasingly risk of extinction, this methodology is starting to be studied worldwide, and there's a need to spread and standardize the methods, as some coral researchers have no experience with conventional microbiology. That's why they have been frequently asked for advice on how to proceed with such approach.
To begin, from each targeted sampling site, use sterile bottles with screw caps to collect 500 milliliters of water in triplicate. If sample processing is happening later, keep the bottles at 4 degrees Celsius. Use a sterile pair of pliers and forceps to cut coral fragments from the same sampling site of the water samples. Rinse the sampled coral fragment using 20 milliliters of sterile 3% saline.